All right, we're here today to talk about DNA replication, so let's get started with that. DNA replication is a semi-conservative process. It's semi-conservative because that basically means that when we have our template strand, which is, we'll say, our original strand of DNA, that's going to separate. And each individual part of that DNA strand, which is a double helix, which means there's two strands of nucleic acids making up our one DNA strand, one template is going to make one new strand of DNA, and another template is going to make another strand of DNA. In order to do that, we need a lot of enzymes working on this piece of DNA in order to make sure the DNA separates appropriately and to make sure that the correct bases are put in. When we talk about the two basic steps of DNA replication, the first thing we need to do is separate that double helix. The two individual nucleic acid strands need to separate from each other. Those hydrogen bonds have to break, and then new base pairs need to be attached, and that's going to happen with another enzyme. So we have an enzyme to unzip the DNA, and we have an enzyme to put new pairs into the DNA. When we look at replication as a whole, this is going to start with a protein that's going to attach to the DNA, and that's going to have something called DNA polymerase that's going to help attach to it. And where that's going to attach is what we call the origin of replication, or the ORI. We talked a little bit about an ORI when we talked about binary fission earlier in this book, and we were talking about how the prokaryotes replicate, and we said the ORI was where that replication started. Well, that's exactly where we're going with, because the only time we're doing replication is when we're getting ready for cells to divide. And that's because we have to have one copy of DNA for the new cell, for each of the two new cells, the two new daughter cells. So let's look at a prokaryote first. A prokaryote is going to have one circular chromosome that's usually going to be in the nucleoid of that prokaryotic cell. The replication for this cell is going to begin at the ORI, or the origin of replication, and that's going to form two replication forks. A replication fork is the area where the double helix has sort of been separated, but there's separation, but part of the double helix is still connected together. Kind of looks like a V or a fork in the road, which is why it's called the replication fork. So there's going to be one fork going each direction in this circle of DNA that the prokaryotes have. Now this process goes extremely quickly, about 1,000 base pairs a second, which is stupidly fast. And this takes place with DNA polymerase attaching to the prokaryotic DNA strand and just sort of going around in each direction. You'll see that the arrows in the pictures are going in different directions, and we'll talk about why that happens in a little bit, but that has to do with the arrangement of DNA. Now, prokaryotes do have a feature where once one strand has started to replicate, each of the new strands that are being created might start a new ORI on each of those before replication is even complete with that first original strand. And that's just to speed up prokaryotic division, because prokaryotes will divide anytime the conditions are appropriate for them. With eukaryotes, we kind of have a different chromosome shape. We have long linear chromosomes. In humans, we have 23 pairs of long linear chromosomes. And in order to speed that up, because our chromosomes, whether a prokaryote has thousands of base pairs, eukaryotes are going to have potentially millions of base pairs in each chromosome. So we're going to have multiple ORI along each individual chromosome, and those replication forks are going to work their way towards each other, and that's okay, because that's going to speed the process up a lot for us. Now, if we are getting this whole process going, it all starts with something called primer. And primer is similar to the stuff when you paint a house. You're supposed to put primer on a wall first, and then you put the paint on the primer. If you just put paint on the wall, a lot of times you'll see spots in the background. The primer is something that's going to kind of make the color a little bit more uniform and make it stick better. And primer is just individual bases that get attached at the beginning of replication that are going to allow the DNA base pairs to actually attach to it. That primer is eventually going to be replaced with regular nucleotides, and that's a job of one of the many DNA polymerase that's going to act on it. So when we are looking at this DNA strand, we see some numbers on this picture. We see a 3 with an apostrophe and a 5 with an apostrophe. The apostrophe is read as 3' and 5'. The template strand is that original strand of DNA that's been separated. The template strand is always read in 3' to 5' direction. Now, one thing we need to know about DNA is that it runs anti-parallel. So, on the 3' end of one strand of DNA is going to attach with the 5' end of the opposite or complementary strand of the new DNA. So that new strand that's going to be laid down on that template is going to be laid down in the 5 to 3 direction. This does get a little confusing for people, but it's just something you got to remember, that the template is always read 3' to 5', and the new strand is laid down in a 5' to 3' direction. And that has to do with counting carbon atoms in that deoxyribose sugar that makes up the nucleotide base, or the nucleotide, excuse me. And the 3' is the carbon atom that is on the sort of, I always consider it kind of the bottom. It's the flat surface of the nucleotide or the deoxyribose in the nucleotide, and the 5' carbon is the one that attaches to the phosphate group and is on the pointy side of that deoxyribose sugar. So we read in 3 to 5 on the template, we lay down the new strand in the 5 to 3 direction, and that's so that these strands are anti-parallel. So if we were to separate them, we can make exact copies of them. Now, DNA polymerase is the enzyme that is going to create the DNA polymer. And we have a bunch of different ones of these inside our cell. It's a pretty big protein complex that lives in the nucleus. If you look at the picture marked A, you can see how much bigger the DNA polymerase is than the DNA. The DNA is represented by the red backbone strips and the white strip that represents where the nucleotide bases would, or nitrogenous bases, excuse me. The blue part is all more areas of the DNA polymerase, and it's a big molecule because it's got to sort of wrap around the DNA, and it just sort of runs down. I always think of it kind of like DNA polymerase is kind of like a hot dog bun, and the hot dog would kind of be like the DNA running through the middle of it, except DNA would be a ridiculously large hot dog. Now, when we talk about replication and talk about that with eukaryotes, we're always going to talk about leading strands and lagging strands. And the easiest way I can describe this one to you is, remember, we said that the template strand is read in the 3' to 5' direction. Well, when you look at the replication fork, the one that goes 3' away from the replication fork and 5' towards the replication fork is always going to be your leading strand, and DNA is going to continuously move towards that, because at that replication fork it's going to keep opening up, and new DNA is going to be opened up to allow for bases to pair to it. So that leading strand is going to just continuously grow. It's not going to have any stop points. The lagging strand, well, remember, we lay down primer first, and then we lay down the new base pairs, and that primer will eventually get replaced. Well, as the replication fork grows, that there's going to be some empty bases, and then there's going to be new primer laid down, and DNA is going to catch up. And if we look at this picture, hopefully we can see this. On the top picture, we see little primer on the very top in the green, and then the base pairs is going in the red, and the reds are going towards that replication fork, and that's going to make that top strand the leading strand. In the second picture, we can see that the replication fork has moved farther to the right than where it was. And we look at the bottom picture, that replication fork keeps moving to the right. Well, in the middle picture that we have here, that primer where it started, all of a sudden the replication fork is way farther away than where it started. So on the bottom picture, there's a new primer set, and then more red bases are being added until it gets up to that individual green segment of the first primer that was laid down in the second picture. Each one of those chunks of DNA is what we call an Okazaki fragment. And Okazaki fragments are the pieces of DNA, or the chunks of DNA, that are laid down on the lagging strand. There's always going to be a gap between Okazaki fragments because we don't attach the fresh DNA onto the primer. We don't want to do that. So eventually that'll get fixed. There's going to be another enzyme that comes through called DNA ligase that's going to bond those two things. That bond that holds the nucleotides together in the same strand is called a phosphodiester bond. And DNA ligase is going to catalyze that bond. But those Okazaki fragments are going to have that small gap that's going to require that DNA ligase to come through. So if we look at this picture, this is sort of going over Okazakis. As we can see, the 5' end on the template strand is on the left, the 3' end is on the right. And that means that the replication fork is going to be to the right because we can see the arrow of the new strand that's being laid down going from right to left. Well, RNA primer is being laid down on the top picture. RNA primer moves off of that template strand, and then as we move to the bottom, DNA polymerase adds new bases. And they go all the way up to that new chunk on the new strand, all the way up, and they leave that small little gap. Well, once it gets there, that DNA polymerase is going to cruise off, and a new DNA polymerase is going to come by and replace the primer with base pairs, and now we have a gap between the base pairs of the Okazaki on the left and the base pairs of the Okazaki on the right. That DNA polymerase is going to leave the DNA then, and a DNA ligase is going to come in, and that's going to catalyze our phosphodiester bond. Now, this is a lot of stuff going on, and realistically, I just told you exactly how DNA is replicated already. Even though it went kind of quick, we unzip the DNA polymerase comes in, lays down complementary base pairs, and hopefully remember our complementary base pairing rules. Adenine always pairs with thymine, guanine always pairs with cytosine. We always have a pyrimidine, either guanine or adenine, pairing with a pyrimidine. I may have said that backwards. A purine pairing with a pyrimidine. Adenine or guanine is always going to pair with thymine or cytosine, and that has to do with the different bonding sites on those nitrogenous bases that's going to hold the two DNA strands together. So, how does all this sort of get protected? Because sometimes there's some mistakes, and in the next presentation we'll talk about mutations and all of that crazy stuff. But when the fragments are completed, there will be some DNA left at the end of the strand, and that's because there's going to be primer at the end that's not going to have anything to pair with. And eventually, that's going to cause our DNA to get shorter. So the older our cells get, the more often they divide, the shorter they actually become. And when DNA gets damaged, ideally a DNA polymerase will come by and fix it. If there happens to be a break, DNA ligase can create a phosphodiester bond. But sometimes, as chromosomes are getting shorter, or if they break and they start to attach to each other, our cell has to have some kind of defense, because we don't want the ends of the chromosomes to just completely shorten up and get into the coding portions, which we call the genes of our chromosomes. Well, we do have a little bit of a defense mechanism. And that defense mechanism that we have is called a telomere. A telomere is a repeating sequence of DNA. In the case of humans, we have a bunch of TTA-GGG sequences at the end of this. And it could be hundreds or even thousands of these sequences at the end of each chromosome. Well, basically what this acts like is kind of like the ends of your shoelace that everyone always tells me is called an aglet. And the shoelace end that has that little plastic piece around it is there to make sure the shoelace doesn't fray. If you've ever broken that part off of a shoelace, all of a sudden your shoelace gets shorter and shorter. So this telomere is there to sort of protect it, because when we have that little end sort of hanging off, that can be clipped off. And our DNA does actually kind of get a little bit shorter over time. We have this gap that gets created because the primer degrades and no new bases are added to that portion. So those telomeres just sort of get lopped off, and since they don't code for anything, it doesn't really make any difference in how our cell is going to function. Now, if we were to shorten off some of the genes and chop some of those off, then that would cause some problems, and we'll talk about that another day. Now, we said that DNA base pairing happens really quickly, and we're talking 1,000 base pairs a second. Guess what? Every so often, mistakes are going to happen. So we need to have a little bit of a defense mechanism, or even kind of a proofreading thing, kind of the same way you do in your English classes when you write an essay. You write it out, then you reread it, and you say, hey, we got a problem here, we need to fix that. Well, we have DNA polymerases that go through and reread our DNA after it's been laid down in what our cell initially thinks is the appropriate order. If it finds a mismatch, it's just going to take that thing out and kick it out. As we see in this picture, as it goes, it proofreads and does stuff. But once it's done, it can still do it. This picture is showing on the left, the yellow base pair is the incorrect base pair to go with that blue one. So as the DNA polymerase is going, it's like, uh-oh, this thing's no good, boots it out, brings in the red base pair that is appropriate, and that goes well. In this case, all of a sudden we're reading, we say, hey, we got a mistake here. We have a yellow one where we should have a blue one, and that's just the colors. Our DNA isn't really yellow or blue or red or green. It's just a molecule. This picture is just to sort of highlight the mistake, and DNA polymerase will come by and take a segment of DNA and replace that right there. So that's kind of like spot checking after you're all done. Now, something that scientists use to amplify DNA and make replication happen really quickly is something called the polymerase chain reaction, or PCR. This is something where we take a strand of DNA in a lab and we amplify it. And when we amplify it, we make a lot of copies really, really, really quickly. And this happens because we use electric currents, we use high temperatures, we use some salts, we use some other buffers and other chemicals to make it so that DNA will amplify really quickly. Well, the reason we do this is so that scientists can study certain aspects of DNA. I don't go crazy into the actual process of PCR. What I do want you to know is that PCR is a way to take one copy of DNA and amplify it and basically exponentially replicate that DNA. So if we wanted to study a specific segment of DNA, which we'll be talking about in some labs that we do, and we do electrophoresis and everything, we could potentially take a segment of DNA, amplify it a bunch of times, or run it through PCR to amplify it, and then we can study some good stuff in there. So I hope this clears up the replication process for you, and I hope that answered any questions you may have. We'll be talking about that in class, and next slides will be all about mutations. Have a good day. Thank you.